Welcome to the first San Diego Digestive Research Center microscopy seminar. Together with my great colleague, Dr. Ken Kim, we offer services in our histology core. So we do have office hours for SDDRC folks. If you have any questions about pathology, you can click on this and get connected with Ken every third Thursday of the month. And I have my office hours actually tomorrow. So if you're going to have some follow-up questions about this, please join us. So we will start our presentation, and that presentation, as well as links to any publications mentioned here, will be sent to you all after I have some time to put it all together in a neater form. But welcome. Thank you for coming. We're going to be talking today mostly about fluorescence imaging. You may be familiar with bright field as well. That's kind of the classical microscope that you can think of. It is the microscope that you would use in your tissue culture room. It is usually in true color, which means that if you're looking through the eyepieces of the microscope, you're going to be seeing very similar image camera. With the fluorescence imaging, we typically use grayscale as our mean of recording information. For bright field imaging, we usually have, this set of beautiful classical histological stains, immunoperoxidase methods, the early days of immunolabeling in tissues. This all belongs to bright field. And on your computer, those images are formed from 3 channels, red, green, and blue. All of those channels on your computer are actually in an 8-bit format. We're going to talk a little bit more about bit depth. That's a kind of an important topic. In terms of the fluorescence imaging, those images can be in 8, 16, or 32-bit format. We can much more easily multiplex our channels. Pretty much any new technology that's being developed in microscopy is developed in fluorescence imaging. And because of that, we're going to be focusing more on that today. So it wouldn't be a microscopy seminar if I didn't show you a demo of how fluorescence actually looks like. So we're going to first be looking at tonic water that I have here. And we're going to be using 405 nanometers light to excite tonic water. This is the demo that was the first observation of fluorescence in human history. And the guy who saw it first described this color as celestial blue. His name was Herschel. Adding gin to that mixture does not spoil the fun. This can be an interesting party trick. Just be careful about that light. 405 can actually damage your eyes over longer periods of time. Over here, I have a bottle that has 70% isopropanol inside. As you can see, the background fluorescence is pretty minimal. There is literally nothing that glows. And I'm gonna add some turmeric to that bottle. It contains curcumin, and curcumin, when dissolved in alcohol, has a pretty interesting fluorescence profile. Pretty cool party tricks for you. As you've noticed, those colors are different. They are different to your eye. They will be different to the camera. That's the basis of multiplexing. And we're going to be talking a lot more about how you can multiplex different channels for different biological information on our next session. You've noticed perhaps that in the chemical structures of both quinine, which is contained in tonic water, and curcumin, you have those aromatic rings. In many fluorophores, you're going to find this. This creates like an antenna that allows photons of light to be absorbed. Electrons in those molecules get kicked into a higher energy orbit. We are then observing continuous relaxation of those molecules to a lower energy state. And what is pretty important is that the excitation light is always going to have higher energy than the emitted light. The guy who first discovered this, his name was Stokes, And he actually had a pretty interesting system for observing that. So he was sitting in a church. He used the stained glass window as his excitation filter, and as his emission filter, he was using a glass of yellow colored wine. With simple means like that, he was able to formulate something that we know as Stokes law or Stokes shift, as we call it in microscopy textbooks. That's observation that you're always gonna have your emission at a longer wavelength than excitation. So at LGI, we can image anything from the level of the whole organism down to a level of subcellular details. And our great cryo-EM core microscopy facility can image all the way down to a level of single atoms. In light microscopy, we are doing a lot with histology and slide scanning, but I want to show you first a couple of things about non-invasive imaging. We have an instrument called AMI-HT that we're using for that. I'm going to use this as an excuse to tell you a couple of things about how microscopy and how detectors for microscopy work. As I mentioned, we do a ton of histology and slide scanning. We use an automated slide scanner, Zeiss Axio Scans U1, that can do bright field and fluorescence. And we also have this simple and easy to use Keyence VZX microscope. We spend a lot of time doing intravital microscopy, and we also do a lot of live cell microscopy. This is an intravital recording from mouse liver that has been blasted with a 2-photon laser. We made a little hole in that tissue, and those green cells, neutrophils, are flocking to that site of damage. It takes them about half an hour to sense that damage, and then they cover that wound area almost entirely within an hour. We use Leica SP8 for that, and for our live cell microscopy, we use our inverted confocal microscopes. And those inverted confocal microscopes can be used for enhanced resolution confocal microscopy. Over here, we're looking at a piece of human pancreas that has been stained with insulin and its precursor protein proinsulin. Probably the most interesting aspect for most of our users is our multiplexing capabilities and our advanced imaging analysis. We have Fluorescent Orion, that's a spectral slide scanner that can detect 18 channels, and we pair this data acquisition with image analysis in open source software QuPath. Let me start with luminescence imaging. These days, you can actually buy petunias that have firefly luciferase gene inserted in them, and they do glow. You have to adapt your eyes to darkness for a couple of minutes, but once you do, they are actually pretty bright. The advantage of luminescence imaging is that while it's weak, it almost has no background. So it has been traditionally used as a very sensitive method of detection of transferred tumor cells or immune cells. And we can use enzymes isolated from organisms such as firefly or urinilla. There are also some enzymes that were engineered based on those. And the important thing that the substrates, the things that we need to give those enzymes so they can glow, are usually very well tolerated by the animal. So we can do longitudinal imaging with those constructs. And they were traditionally used in many immunocompromised animals for monitoring tumor growth, but that's the key, immunocompromised. And this is different than any other microscope or imaging device that we have. Most of those imagers are absolutely calibrated, which means that you actually get to see the number of photons that you have recorded on your camera. And because of that, we don't really want to use the uncalibrated number of counts for reporting of our data. We want to use calibrated units such as radians or total flux. As the tumor in the animal will grow, you actually need to make sure that you don't saturate your images. And this is like the single case where it's absolutely fine to change your exposure settings because that system is absolutely calibrated. So saturation is always kind of the worst thing that can happen to you, because once you hit that ceiling, you're not able to know how much brighter this could have been. If you transplant something from a firefly into a mouse, you may expect to get some immune response. And that problem has been known for quite a while, but it's not well known to people who are just starting with that kind of imaging. So I put a couple of references here. This can also happen to transferred immune cells if they express GFP or other fluorescent proteins. That problem is typically seen in longer-term experiments. If you are doing something very acute, you're probably not going to give the mouse immune system enough time to really respond to it. And of course, you can use immune system to fight against immune system. So if you express your antigens in thymus, you will achieve central tolerance in the animals. Those are two examples of papers that show you how you can tolerize your mice to your transferred antigens. This may not be a problem in your system. It depends on the background of the animals and other things, but it's an important consideration. So we have a first interesting use case. This paper came from one of the SDDRC labs from Christina Lorente and Bernard Schnabel, where they designed this acute in vivo killing assay. They had bioluminescent bacteria that were injected into exposed loop of jejunum. And in the case of this bacterium that they were studying, they noticed that this particular mouse knockout is doing much better job at killing those intraluminal bacteria. That's why they're still glowing after, I believe, an hour here, compared to wild type mice. And their control, E. coli, did not really show that preference. So what we tried and failed back in the day was that we were trying to do long-term monitoring of infection with Salmonella. And it turns out that the signal that we were getting was much more correlated with the amount of abdominal fat that the mouse had than with the amount of bacteria. And this points to something very important in those experiments, and that is the shadowing or light blocking effect. Light propagates in tissues in an interesting and not necessarily straightforward way. So if I asked you, where do you think the signal is located in this phantom mouse, you'd probably tell me that it looks like it's located in the lungs of that phantom animal. But if we flip that mouse upside down, you'll actually notice that the very strong signal is probably coming from behind the sternum. So because we have so much scattering and absorption in mice and other animals, we cannot really precisely pinpoint the exact location where that signal originated. If you're ever going to be doing that kind of imaging, it is a very good idea to shave or depilate the area of interest and essentially just flip the mouse to make sure that you're indeed looking at the brightest signal. A very good idea is also, after you conclude your experiment, to isolate individual organs and image them to pinpoint the location of the transferred cells, or in this case, we were studying distribution of near-infrared labeled nanoparticles. And you can see that they are really glowing in the lungs, spleen, but not so much in the liver. That brings me to the fact that there is this phenomenon of biological optical window, and I can demonstrate this to you by putting bright white light behind my hand. You're only going to be able to see red light passing through. The green and the blues are absorbed. That's because our blood absorbs and attenuates a lot of wavelengths over here. By the way, our firefly luciferase emission is happening here. There's a bunch of papers in the field that show that you can have something that's brighter, like Ranilla luciferase, but actually the light from that is going to be absorbed to a higher degree. So your mileage may vary. You actually have to try it both and see which one is going to be better. So when doing detection of nanoparticles, let's say, or some constructs that you've put into the animal, it's really good to target them with near-infrared fluorophores. And the other reason for doing that is that the autofluorescence that's inherently present in the animal is much less severe in near-infrared region, and you can further knock it down if you keep your mice on diet that does not contain chlorophyll, alfalfa-free diet. So let's take a look at the imager itself. This is how it looks. And if we take the hood off, we're gonna see a very prominent camera on the top. So that weak signal is detected with a very sensitive CCD camera. And what's important about this chip is that it has large dynamic range. We understand large dynamic range as our ability to measure both signal that's weak and strong. In order to do that, that chip needs to have very low inherent noise and very large capacity for holding small amount of signal, but also allowing you to see a lot of signal. And what really helps is if that chip would have a large bit depth. And bit depth is just how many gradations you have on the scale. So if this is a pixel on my camera, I have it filled a little bit more than halfway full. I could read it out in my computer in an 8-bit mode with values from 0 to 255, or in 16-bit mode with values from 0 to 65,000 with change. More gradations really help with quantitative imaging. What we want to do when we are setting up our instruments for imaging, we want to make sure that we'll set it up in a way which will allow us to capture unexpectedly high signal, but also see the low signals. And, you know, with a lot of biology, you just don't know. But we typically want you to set up your instruments with the highest possible signal, like your positive control. And that hopefully will allow you to capture all of the variability in the signal that you have across your samples. The trick to this is that this chip is actually cooled, because sometimes your exposure time can take up to 10 minutes. And that brings me to the point of noise or uncertainty in microscopy. We take care of that thermal noise by cooling the camera to minus 90 degrees Celsius. Anytime you read something with electronics, you observe this read noise. If you multiply something, for example, with a photomultiplier tube, as we use in confocals, you're going to get some multiplicative noise. But the most important and often most dominant noise in our imaging systems is photon noise. And I'm going to let Jennifer Waters from Harvard Medical School Microscopy Corps produce herself here and tell us more about the SNR, the signal to noise limits. I just want to plug an image analysis and imaging course that she hosts, as well as the series on YouTube called Micro Courses, from which I took this video, and an online microscopy training micro tutor.
So why is there noise in our images? When we snap an image of our specimen with the microscope detector, for example a camera, we are counting the number of photons emitted by our sample that arrive at the detector during the exposure time. Now, if photons marched in perfect order, we would get the same count for each exposure time. But the photon flux is stochastic, and so the count that we get differs between exposure times. This variance is called Poisson noise, also known as shot noise. Poisson noise is a fundamental limitation on the certainty of measurements of intensity.
You can go to the Micro Courses website, and there is also this wonderful book by Pete Bankett called Introduction to Bioimage Analysis. And there are those courses called Micro Tutor courses, very good online learning resources that cost you exactly $0. If you want to spend between $4,000 and $5,000, you can and you should go to quantitative, analytical and quantitative light microscopy, or this quantitative imaging courses. They are fantastic. For me, AQLM was my microscopy education was before and after that, right? This is just like something completely different. For 10 days, you are spending time with super smart people that show you how microscopes are built, how everything works. It's from 8 a.m. to midnight for 10 days, okay? It is super intensive, but it's a lot of fun. It is fantastic. Highly recommend it. This is taken from Pete's book. Before I tell you how we can fix that problem with photon noise, we need to talk a little bit about our images. On the computer, they are composed of pixels or picture elements, and pixels are really numbers. We just display them in a form that's useful for us. And one of the things that we do is we use something that's called lookup table. And that essentially is a table that determines what color we're going to see on the screen when we have certain value of a pixel. And we can change that lookup table, and we can change the colors or the brightness of the images that we see. We don't actually change the underlying data. We just change the visualization on the screen. And that's important because I told you that we would like to acquire our images, at least in some cases, in 16-bit mode, but our screens are 8-bit. They only have values from 0 to 255. If you were careful enough, we just moved from something that has 10 centimeters to a cell that has a little bit over 10 microns. We're gonna talk a little bit more how we achieve that. I would like to introduce you to this image histogram. It's something that you're going to find almost in every microscope system. And it's going to be very important that we understand how we can adjust it to make better use of the data that we are recording. So on this axis, we have signal intensity, and on this axis, we have number of pixels. What those two things are doing, the black and white levels, that's essentially mappings. Pixels of that intensity will be mapped to complete black, and pixels of that intensity will be mapped to completely white. And anything in between is going to be shown as grayscale. So what follows is that this large number of pixels here represents our background in the image, and that is our signal. So if you want to increase your signal to noise ratio, you need to collect more photons. This is an exposure of 1 microsecond. If we use the same display parameters and increase the exposure time by a factor of 10, we're going to see an image that's not very useful, right? It's too bright. We cannot really see the detail. So instead of keeping the display settings up here, I'm going to stretch that contrast, and I'm going to adjust it. So now I can see much more useful image on the screen. I think we agree that this image looks noisy, right? It looks grainy. Some of the features here are not well visible. Our structures are lost in the noise. And if we go for 100 millisecond exposure time, although that image looks similar on the screen, our pixel values are very different, okay? So it is important to make sure that we both look at our image histograms and our images. And at the very end, I'm going to tell you how you should be presenting your images in your figures and publications. And essentially, you want to make sure that you adjust those values so they are the same across your panels in the figure. We can show this in a different way. We can create a line profile across this line, and you can see that this little blip becomes so much more visible when our signal to noise rises. Increasing the intensity of the light source or increasing the exposure time can help with this. But this will not replace your good sample preparation, and that's why we have the next session next week on the little bit more details of how that sample preparation can really be perfected. So we moved from centimeters to microns, and we did that with the help of an objective. When you think about objectives, you think about magnification, and that's definitely a good first impression. But what I would like to focus your attention on is a second number that's given after the magnification of the objective. That's so-called numerical aperture. And that's really the heart of the magic that happens in the objective. When you're going to be reporting your materials and methods, stating just the magnification without stating the numerical aperture is not going to be sufficient, because the NA drives all of the characteristics of the objective so much more than the mag. And when you have higher numerical aperture, you're going to collect more photons, you're going to have more resolution, you generally have lower depth of field, and you're generally going to have much lower working distance. The choice of the objective sometimes is dictated by the sample that you have, but if you want to have more resolution, you do want to use higher numerical aperture lenses. And there is this interesting phenomenon that you only get to kind of think about it more when you're embedded in that intensive microscopy course for a couple of days. But that fluorescence originates in your sample, but it spreads in 360 degrees, right? There is nothing that tells photons, hey, move down here, there's an objective looking at you below. So you're effectively capturing just a small fraction of the light that's being generated at your sample. And the bigger the NA, the more of those photons you can collect. Now, the quirk thing about optics is that if you take a single point of light and project it through optics, is not going to be a point. It's going to be something that resembles an airy disk. And there is a mathematical formula that we can use to describe it that's called point spread function. And this is a point spread function on one of our objectives in a microscope, essentially a blob. And if you are not gentle with your objectives, you're going to whack them. That point spread function is going to be more wacky. and all of the subsequent imaging that you're going to be doing with this objective is going to be flawed. So it's extremely important to take good care of your objectives. And if you're coming to any training with us, you're going to have plenty of opportunities of learning how to do it right. So those extremely expensive pieces of equipment can be safe. To put it in another way, image of a point is not a point, and that's the point. What follows from this is that if you have a crowded sample with lots of fluorophores, and that's every sample that you're going to be looking at pretty much, those crowded airy disks blend together. And that's why you cannot have unlimited resolution in your image. Resolution in light microscopy is going down to around 300 nanometers, and that's a lot. That's like 30 times the thickness of the plasma membrane. It's just a fundamental physical limit that you cannot break, but you can go around it, okay? And those are some of the techniques that people developed to go around this problem. The one that we are using most often is called airy scanning, where we take information about this whole airy disk pattern and we reassign photons to increase resolution. You can think about a way where you will eliminate only certain parts of the image, and then not every fluorophore is going to be excited at the same time. And those microscopy methods are called SIM. You can create conditions in which some of those fluorophores are going to go into so-called dark state, and they're not going to be visible. And those methods are called storm. You can do some fancy engineering to the point spread function itself and shape it in such a way that you excite much smaller space in your sample. And those methods are STAT. Or you could look at fluctuations of molecules over a certain period of time, and that gives you SURF, super resolution radial fluctuations methods. So there are many different ways how we can go around this fundamental problem, but it will exist. And very important from a technical point of view or practical point of view is that you can have the best microscope with the highest resolution possible, but if you set pixel size to be very, very, very big, what you're going to get in your computer is an image that will not actually show you all of that resolution. You have to set your pixel sizes to be small enough in order to capture that. Now, you don't always need to work at the highest resolution. This is one of the trade-offs, one of the compromises that we teach you when you are doing training with us. It really depends on what you need to achieve, and you're always going to be compromising in microscopy. You can take the most detailed image, but it's going to take you time. It's all about making trade-offs. A lot of the systems that you may have initially worked with are so-called wide field microscopes, non-confocals, as we can put it. And they are great. They're fantastic. They're very light sensitive for flat two-dimensional samples. But when you look at your sample with a high resolution objective, you're going to see that there's a lot of blur that's coming from out of focus parts of that specimen. And if we look at the So this is a three-dimensional rendering of a Z-stack from a wide field microscope, and you can see this hourglass pattern, which makes it very difficult to interpret your data. Wide field microscopes are extremely fast. One frame took only 15 milliseconds to acquire. On the other hand, confocal microscopes, the same cell is shown here. It takes a lot longer to acquire your image, 4.7 seconds. For a lot of uses in microscopy course, confocals are really workhorses because you can get that level of detail and that level of contrast, and that's really why we have 3 confocal microscopes in the core, and only one of them has wide field capabilities. So meet our inverted confocals. We have Zeiss LSM 780, 880, and 980. Those 2 instruments are equipped with airy scan detectors that does this pixel reassignment, that super resolution. They can be used for a variety of different techniques. What we are showing here is an example of live cell imaging that was done when we were looking at killing of tumors by engineered CAR T cells. So here we have our target. That's a CAR T cell, brief interaction, kiss of death, and that cell starts to pick up propydium iodide because it is dead now. The newest baby has some improvements in navigation, has AI sample finder that helps to find the sample. So I wanted to show you this fun experiment that was done by one of the labs here, Jingru Fang, and from Erika Ullman-Saffer lab. She was studying viral replication factories. They were looking at Ebola virus, not at the exactly fully functional and able to kill you Ebola virus, but at certain proteins that were cloned from it. So we have VP35, we have NP, and we have L protein. And what happens in this experiment is that you can select a certain area because it's a laser scanning microscope. We can actually focus the laser on a particular spot in a cell, and we can bleach the fluors that are in this location. So you start with your intensity and then you bleach it, and then you can see how quickly that signal recovers in different conditions. So that's a nice technique that can show you the internal dynamics of molecules in your cells.
Okay, hi everyone. My name is Laurence Cagnon. I'm the Biosafety Officer for La Jolla Institute. And I just would like to review a few safe work practice when you are going to bring samples to the microscopic core. One of the things that you need to be clear with the microscopy staff is what type of samples you are going to bring. So you need to clearly communicate what cell type, if they are infectious or not, and if they are fixed or not. So just so you know, all the human cell lines we are working with in the laboratories, like 293 cells, HeLa, THP1 cells, they are all biosafety level 2. And they need to follow some guidelines to be handled safely and not put other people at risk. What we want you to do once you've explained what your samples are is to move your samples safely from your lab to the core. For that, you need to seal your dishes, and you can use paraffin or tape. And then you are going to put them in a secondary container that needs to be closed tightly. So in case you trip, you are not going to spill your samples in the corridor and create kind of a hazardous spill. So the other thing is that if you are going to be working with something infectious, you need to fix the samples. That's what we would like you to do. So in order to fix the sample, you need to use validated fixation methods, but you can also have different methods, but they need to be validated in your lab. And you need to communicate with the core what inactivation process you have used, so they can verify also on their side if they feel comfortable with that. If you have any other questions or concern on how to prepare your samples for the microscopy core, then you can contact us at EHNS and we'll help you prepare your samples. If you need to work with live cells that could be causing a health issue or concern, we will also help you in conjunction with the core staff to establish a process that is safe for everybody.
We do have scopes in BSL-2 facility. Those are mostly used for imaging animals. We have intravital microscopy capabilities here, but we can also image some infected samples on the scope. So this is our multi-photon and confocal microscope. We have a lot of surgical equipment and tools to do intravital imaging. We can measure cell movements. It's also used for looking at organs in 3 dimensions or clear tissues. This is an example of an image of a mouse small intestine that has been stained with phalloidin. And that mouse received DSS in drinking water, and you can see those villi that were damaged in that process. And this was a Z-stack that was taken on this microscope, and then we made maximum intensity projection to kind of show you different aspects of the morphology of those villi. But I think the this is one of the favorite papers that use this microscope that I really like for the way how it was designed. People from Shane Crotty lab here wanted to study the effects of valency of antigens. So you can have a monomer, you can have 4 pieces of a protein, you can have 8 pieces of protein, or you can have 60-mer. They immunized mice that received a couple days before B cells that were either specific for that antigen or were polyclonal and just didn't care about it at all. And what they were looking at is the movement of those B cells in the lymph node, the fraction of cells that were arrested, that stopped. Because while we did not visualize the antigen in this case, we could infer whether or not these cells come in contact with antigen-presenting cells by looking at their movement. So let's take a look. Cells are moving around in an immunized animal, and you're gonna want to watch the green cells. And now there's gonna be a protein that cannot be recognized by the cells, and the green cells started to move again. Let me play this once again so you can appreciate it. So green cells are moving, and as the valency of the antigen increases, they're gonna be slower and slower and slower and slower. Whereas the blue cells just don't care because there is nothing that they can respond to. So the most important rule of intravital microscopy is that you really want to take care of your animals. And we published this protocol on looking at intravital imaging in the gut. And our guiding principle and your guiding principle should be to make sure that you induce the least amount of stress on the animal. It's really the goal, because if you're going to be looking at animals that are stressed, injured, damaged, hypoxic, unperfused, and photodamaged, you're not really going to be able to glimpse at that behavior of cells in their natural environment. Those are some movies from our intravital recordings in the gut. The red cells are intraepithelial lymphocytes that are migrating in between epithelial cells in the mouse villi. And we can also visualize this on this color time scale. So each color represents different frame in the video. And in the case of wild-type animals, we see that those cells are moving pretty quickly. And in the case of animals that were knockouts for HVAM in enterocytes, those immune cells are much more stationary. And we can also visualize this on those so-called spider plots, where you can see individual tracks of cells over the time frame of the movie. And in the HVAM knockouts, they are staying still a lot more. And that's another measurement that we could derive from those videos. This is the area that's covered by those moving immune cells over a time frame of 10 minutes. So other examples of biological questions that were addressed with microscopy methods, we were looking at behavior of neutrophils in sepsis. In those conditions, neutrophils that roll on blood vessels inside mouse body, they can actually form those very long tethers, as they're called, and some of those tethers can break off. And interestingly, you can also find those structures in blood of septic people. We created those flow chambers, and we had an assay where we had human neutrophils rolling on the substrate, and we were able to show that under certain conditions, those neutrophils also can form those ends, as we call them. And this is a movie where we are increasing flow rate, increasing shear stress, and this shows you image analysis and quantification of those different types of tethers or ends. And here we have graphs showing that when neutrophils roll on p-selectin at above 40 dynes per centimeter square, you start to reproduce those ends. So quite a bit of cell behavior was studied here. This is an example of imaging in immune cells called neutrophils. We're looking at very tiny mitochondria in them. They don't look alike like in the other cells where those organelles are big and easily visible. Here we had to use Eriscan even in order to be able to see them. Those are those green dots labeled with TOM20. And we were able to measure their numbers and also their area here. That's an example of work that involves slide scanner. Those slide scanners are great because they allow us to see a lot of sample. Here we were looking at type 1D, type 1 diabetes. The researchers were looking at the presence of CD68. This is a marker of macrophages, and presence of antigen-presenting molecules HLA2. In the context of islets that are being attacked by immune system in type 1D, you have a lot of this HLA class 2 in the vicinity of the islets. The numbers of islets that we're analyzing here are pretty staggering. 4,000 islets in control settings, and we had 312 islets in type 1D. There is differentiation between islets that still contain insulin. Those are the ones that are going to be killed pretty soon. And we also have insulin deficient islets here. And that analysis was followed up by confocal imaging on the same samples, where with much higher precision, we could show that these beta cells that produce insulin, they actually express HLA, which would be a pretty clear sign for the immune system. Hey, be interested in me. I'm presenting an antigen that you can see. Probably the most interesting of the new instruments that we have is our Raresight Orion. It's a spectral slide scanner. We have 20 channels. We can acquire 18-plex panels. We reserve some channels for autofluorescence. And it's a game changer for our multiplexing capabilities. What I'm showing you here is a piece of human tonsil. We are looking at a labeling. I'm going to zoom in. And, there is just a handful of channels that we recorded here. And our brains are not very good at, piecing out this information and kind of understanding those rainbows of colors. So I'm going to simplify this to just 3 channels. And we have here CD8 alpha cells. Those are some T cells. There are some markers of dendritic cells, C11C. And there is a marker that can be expressed in the membrane or in lysosome, CD68. What I really like about this instrument is, although it's wide field, it gives you really a lot of details, and it has amazing dynamic range. So you can see those very dim labeling over here. That thing is real. It's just so weak that you can barely see it. And in the same slide, a couple hundred microns further, you can see super bright signal for CD68. I just present it in a way that looks like it's saturated. In the instrument, it's actually not. So that is amazing for us, because for a long time we were able to kind of say yes, no to presence or absence of markers, but now we can actually start to ask questions about how much we have. Analysis of that data is a whole new level of complexity, but we are running a QuPath course, a software package that we use to analyze those images. People come to do this course from around the US and sometimes from Europe. So if you are a San Diegan, I would highly recommend you taking the course, even if you're not doing multiplex imaging or you're not really in that field. It's a great opportunity to be exposed to something completely new, get some skills that may help you in the future. People think this is fun, and we try to make it fun. So consider coming. And San Diego Digestive Research Center covers registration fees. This is not a microscopy pit of despair. This is a way how we design and execute microscopy experiments. We start with biological question. We do experiment planning. We then need to think about our sample preparation, image acquisition, and image analysis. And kind of the point here is that you almost always will want to go back and make a better plan and improve your sample prep, perfect your image acquisition, because now you know a little bit what you were looking for. And that really makes image analysis so much easier. It is a cyclic process that can lead to great data that you can then present to the greater audience. If you want to read more about it, if you absolutely don't want to consult with us so we can help you to design your experiments, fine. There is a paper that you can read that very nicely discusses that process. And you know, one of the very common questions that we're getting is how to know that something is real. In pretty much every other field of science, we try to address that question by doing control experiments. And microscopy is no different, right? So we can help you to design your controls so you can know that what you're looking at is indeed real. So summing it up, which microscope to use? If you're not interested in resolution or are looking at a very flat sample, wide field microscopes, camera-based systems are going to be just fine. But you will often want to follow up with a confocal microscope. If you are using thicker samples, that is definitely a recommended choice. And if you need super resolution details, go with the Eriscan. I will share with you this poster that offers some guidance for quantitative confocal microscopy, going and kind of nicely summarizing all of the steps that one would need to take to have some quantitative data at the end. We're really getting to the end. The lunch arrived and is here and it's going to be served soon. But I want to tell you that when you're going to be ready to publish your research, you want to create figures that are going to tell the whole story. And there are those guidelines and checklists for publishing images. This is a consensus statement of a lot of people who were doing this kind of work for a very long time. And they have some recommendations for you. And you can quickly reference them to, for example, find that when you're adjusting brightness, which you need to do in order to visualize your data, you need to use the same adjustments across all of your images. Okay, something very simple, very useful that gives you kind of best practices that are currently recommended by the field. So with this, I would like to thank you, all of the users of the facility. Just remember that together with Ken, there's 7 of us working in the histology and microscopy and histopathology core. And combined, we have over 92 years of experience. So we very likely will know an answer to your question or will be able to guide you in the right direction. We help with experimental design, sample prep, microscopy, histopathology, image analysis, and a lot more. And we try to create a safe, supportive, and hopefully fun environment for better science. So thank you, and we'll take any questions if there are any.